So thank you, Tony. So I'm going to disappoint you, I think, and tell you it's just fine. I suppose you know, it's uncommon for me not to know what to do with a talk these days, but this is one. I really not quite sure why you were so desperate to hear this talk, but and I sat there and worried about it for quite some time, and you'll see this is what I did with it. It says, I think the title, there were two titles actually, What Do We Know and What Do We Need to Know, and actually they're actually quite sort of separate, different meaning titles, so this is one of them anyway. So there are many generics in the United Kingdom, even in such a highly regulated economy as ours. The triazoles that we rely on, some of these are generics, so voriconazole and fluconazole and itraconazole, and some are still proprietary, posaconazole and isavuconazole. The polyenes, despite amphotericin B being discovered in the mid-1950s, still the formulations are still proprietary because of the complex engineering, and we'll come to this at the end of the talk. The echinocandins, caspofungin, the generics are just available, anidulafungin and micafungin still under patent. And then there are others. Flucytosine, of course, also a very old compound, is proprietary for reasons that I don't really know, but Tom may. Terbinafine, griseofulvin, and ketoconazole as being generic compounds. And so it's really of interest when you look into this a little bit more, which most clinicians, I guess, don't, because they write the active drug up rather than thinking about the specific trade name. And so here's voriconazole. Here are all the agents, all the voriconazole preparations that are available in the United Kingdom. Of course, available means has to survive pretty rigorous regulatory vetting, and I'm going to go through that actually at the risk of sort of terminally anaesthetizing some of you about regulatory science. Actually, it is interesting and important. So here's voriconazole, and then look at fluconazole. The font's much smaller because actually there are a lot of fluconazole preparations, both in terms of the IV preparations and capsules. So there's a vast amount of choice for these two agents, at least as examples. And so I think that this was the reason for the talk. If you search this topic, then there are lots of people in various parts of the world doing bioequivalent studies and showing the PK is or isn't different, and some of them are in quite obscure journals. But this paper from Dr. Pasqualotto when he was with David Denning 10 years ago or so from Brazil reported 3 cases of patients receiving itraconazole and who had generics, and as was David's wont, as many of you know, Roger, he monitored everybody and saw that some of these patients receiving generics had low concentrations of itraconazole, and when he switched them to Sporanox, they did much better. Now, of course, and here are the patients down here. And then this is just obviously it's a letter to the editor, and then the comment is, well, it's fine because you just or the regulators just show that the PK is the same in healthy volunteers, but I treat patients that have low acid, and maybe we should do a better job of this. And well, I'm going to argue actually, and it's going to be my thesis, that actually we do a very good job of this, and we should have confidence in all of those lists that I showed in those previous slides. So here is as someone that's got sort of interested in regulatory science for necessary reasons more recently, here's the relevant document, the legislation that governs us all, at least for the time being. I see that Mrs. May is going to try and keep us all in the EMA, which I thought was a very fine thing to do. And here is the piece of legal hardware that tells us about how generics have to come into the European Union and can be deemed fit for purpose. So 2 medicinal products are bioequivalent if they are pharmaceutically equivalent. So here's a bit of jargon for you, the API is the active pharmaceutical ingredient, so you hear drug developers talk about API all of the time, and in the same dosage forms, or pharmaceutical alternatives, so that's the same active agent but with different excipients. They are the same. From a legal perspective, is the drug exposure is the same following the administration of the same molar dose. And so in the attempt to demonstrate bioequivalence, limits are set to ensure comparable in vivo performance. And that comes back to David's letter to the editor about he was suggesting that these compounds didn't have comparable in vivo performance. And the bridging is done on the basis of the PK. So it's the same strategy that's used to develop drugs for children, Adelia, or neonates. So the PK bridges all the way through. We assume that the dynamics are the same. And that does a good job most of the time, except for very complex compounds, and most notably the polyenes, where, as you'll see, they are all exempt from legislation to enable a bio-waiver, as it's called. So here are the important considerations when the EMA has to make a ruling on whether to license a generic. The physicochemical characteristics of the drug are important, so its solubility, its PK properties, the proportionality and composition, the linearity of the PK, obviously an issue for itraconazole and voriconazole, whether the original license is granted in fed or fasted states. That's obviously important for itraconazole and posaconazole, but fasting for voriconazole. Enantioselective analysis, well, I've been interested lately in Pasqualotto, which is a 50-50 mix of RNS forms, for example. So you have to show that if you're making a new Pasqualotto, you have to have the same amounts of RNS. You have to be able to reproduce that. And you are, there's no point sort of depleting the world of its supply of healthy volunteers, but so you can ask for waivers. So you don't have to do the same study for 50 milligrams, for 200 milligrams, for or whatever the licensed dosages are. And so the legislation's really about oral drugs. So for parenteral solutions, it's interesting that bioequivalent studies are generally not required as long as we're talking about the same API, the active pharmaceutical ingredient, and the excipients, the new excipients in the new generic don't complex and therefore alter or potentially change the PK. And just, I will note and come back to this at the end of the talk, that liposomal formulations, liposomal amphotericin B, is not covered by the bioequivalence legislation, and my cell-forming formulation, so amphotericin B deoxycholate, is also would not qualify for a bio-waiver. I'll come back to these at the end. And so it is possible, and I don't think that this is really relevant for antifungals because of the complexity of the PK, to use simple risk-based tools for a bio-waiver approach. That means you don't have to go and do a formal bioequivalent study. And so these are designed, as is the EMA's want, to reduce the reliance on in vivo studies, and it really is restricted to highly soluble drugs, of which antifungal drugs generally are not, known human absorption and wide therapeutic index or indices, and of course, that if there's one thing that's characteristic about antifungal chemotherapy is that there is not a wide therapeutic index, they're actually exceedingly narrow. And so the BSC is just sort of difficult to develop generic antifungals for that reason. So in terms of the test compound, everything has to be under GMP. And I guess maybe this room doesn't have much exposure to what this means, but it means a lot, actually. I mean, it's interesting. CMC programs really can halt the development of any new antimicrobial. So everything has to be done under strict regulatory conditions. The test product that's brought forward has to originate from a big batch, so it can't just be made in a laboratory and then brought forward. This has to be industrial scale manufacture before it can be brought before regulators. There has to be evidence that the company can produce stuff on an industrial scale. Dissolution has to be established. That's an in vitro study about the way that the excipients enable the API to be disengaged and become bioavailable. And that has to be done on the first 3 production batches. So you can already see that this is a large amount of work. This isn't sort of something that you engage in because, you know, you think you can make a quick buck. It's an involved and rigorous process. Here are the dissolution studies that have to be done. The statistic is for show. I'm not sure what that means, but anyway, it's in the EMA document. You have to show that the dissolution is the same between these two, between the active comparator or the licensed comparator and your new generic. And so here's what, when you front the EMA, here's what they ask. They ask you to match these things: the Cmax, the maximum concentration, the time to the maximum concentration, and various measures of AUC. And there are usually two that they cite. So the AUC for 0 to tau, that's the dosing interval, and also 0 to infinity. And the limits are between 80 and 125% for each of those measures. Again, well, you know how variable these drugs are, Roger. It's not actually an easy task to be able to match anything within such a tight range or bound. And then the EMA is very explicit about the studies that it needs to see for bioequivalence, so that it requires randomized 2-period, 2-sequence, single dose crossovers at the very least. It needs the treatment period separated to enable complete washout to beneath the limit of quantification, generally 5 half-lives. And here is a study plan that needs to be done. So the first is there are 2 sequences, sequence A, B, and then sequence BA. So it would be Sporanox and then the active or the new itraconazole, and then the new itraconazole versus Sporanox, period 1, and then a washout, and then period 2, and with full PK in the middle. So, you know, again, this is not trivial, and it's not cheap either to do this. And the human subjects have to be greater than 18 years old. They have a BMI between 18 and 30. I guess the strategy is to use these healthy volunteers to extrapolate into these special populations that we all treat. That comes back to Dr. Denning's sort of paper or letter again, and that's the point about some have argued this. And I guess the treatment groups have to be comparable in all variables that may affect the PK of the API. So that means pharmacogenetics, weight, gender, ethnicity, and so on. The prandial state has to be addressed, and fed and fasted states are tightly controlled under GCP conditions. The packaging has to be done and done according to GMP, and the company then has to agree to enter into pharmacovigilance studies. So you're starting to get the picture that these are not fly-by-night preparations, or while there's a lot of them, it's just not a trivial exercise. So I just thought that I would summarize for you. I showed you all the voriconazole cases or formulations that you can use in the United Kingdom legally. And so one of those, what was done, this is the relevant document. This was Accord Healthcare Limited. The reference compound, of course, is Vfend from Pfizer at 50 and 200 milligrams film-coated tablets. That was what everything was measured against. Same clinical indications are sought. So you can't make a generic and claim that voriconazole is a good mucor drug without doing the relevant study in mucor showing whatever that would show. So you're forced into the same clinical indications for usage as in the label, and if you don't want to do that, you have to go and do the relevant clinical study. So there was a single bioequivalent study of 200 milligrams of Vfen versus 200 milligrams of the Sporanox. Their PK was within the bound, 80 to 125%. The study was under GCP conditions. This is all documented on the EMA website. It was a randomized single-dose, 2-period, 2-treatment, 2-sequence crossover study, which I had to Google to try and understand, done under fasting conditions because that was what the SPC says for voriconazole. The API had to be strictly controlled in terms of the composition, and I had to convince the chemists that they were producing the same active compound as in Vfend. Batch analysis was provided. Stability was confirmed at various temperatures and times, and up to 44 months. So they had stability data up to 44 months for this new generic. So that's a significant amount of time, and you have to do all of that under GMP. It's not a trivial exercise at all. The dissolution studies were done at 3 pH levels. The manufacturing process was validated, and on those grounds, on the data from the 200 milligrams, a waiver for the 50 milligram tablet was granted. So Tony, I just think that we should sometimes believe in the regulatory processes that many of us don't see, but it's sort of a bit like statistics, isn't it? It's a necessary part of our lives, which many of us are not interested in, but it's pretty important. And I think that I think Dr. Denning's probably wrong here. I think we just have to be very grateful that we have such rigorous regulation, and we should have some confidence about these generics that are available to us. I don't think that there are grounds to sort of suggest that they are somehow inferior. However, I wanted to just think about generics from a global perspective, and I'm very happy to see the good Professor Harrison sit in the back, because I could have chosen one of your studies, Tom, but I didn't. But I don't think the point here is going to be too lost on you. So everyone knows that the generics in India is a powerhouse. So this is just Chennai. These are the generic companies that I was just sort of staggered by the number of generic companies just in a single city in India, let alone in the Indian subcontinent. And so here's the paper I just want to talk about. This paper is well known to you, I'm sure. It was recently published with Thuy Lee as the lead author, a trial of itraconazole versus amphotericin B for HIV-associated talaromycosis, Penicillium malnefii, published in a very prestigious journal, as shown here. And if you read the methods, and I've highlighted in yellow, so either intravenous amphotericin B deoxycholate was purchased from Cipla in India and given at that dose, or itraconazole capsules were purchased from Stada in Vietnam. Right, so here are the results, right? And you know these results, and there's inferiority of itraconazole in terms of the mortality endpoint and also in terms of the CFUs in blood. And here are the potential reasons that you might put to a journal club. You know, amphotericin B is just better than itraconazole. The wrong dose of itraconazole was used. There's a PK failure, so that many of these patients are HIV positive and had TB, and so we're getting rifampicin in the regimen, and of course they all don't have any acid, these patients, so that they had PK failure with lack of absorption, and you know, Roger and I talk about this sort of stuff all of the time. But number 4, of course, is the one that surprised me as I was preparing this talk and thinking about this. There's failure of the formulation, so that Thuy inadvertently used something, well, let me just make it clear that I'm not making any accusations about any of this, but this is theoretical, that she was using something that she didn't recognise, or we don't recognise, as itraconazole. Well, right, so Tom, this is how you do your studies as well. I'd be interested in what your perspective's going to be. So I rang up Thuy. She's now working with John Perfect at Duke. And I did that after I went to the Stada website in Vietnam. And here's itraconazole amongst all of their portfolio, Itrastand or whatever it is. And this is what was used in the clinical study. And then I sort of looked around on the web for generic preparations of itraconazole, and there's a staggering 210 global preparations of itraconazole that are available in the world. You know, and I was putting them on the slide, and there were too many of them, so I just, well, I had to count them up as so it's approximately 210, because I may have miscounted 1 or 2 here and there. But there's the website for those of you who are interested. So the point that I just want to make, and I'd be very interested in the views of others, so the trial is funded by the Medical Research Council and differed, and it's published in the New England Journal Medicine. It's done under GCP. We recognise that this is a very rigorously conducted clinical trial. Yet it's not possible rather to be completely sure about the formulation that was used. And this is usually controlled very tightly, in our own case, by regulatory agencies, and that, like I say, we have a great deal of faith in, even if we don't, you know, like them all of the time. So I rang Thuy and I said, well, did the funders ask you for any statement about whether the itraconazole preparation, you know, where's the bioequivalence studies? And she said, no. Did the ethics committees ask you? No, they didn't ask. I emailed Stada to get that relevant information about bioequivalence and didn't get a reply. And of course, point is that actually I have all the PK for this, and it's a very beautiful data set, but it won't help distinguish PK failure from a pretty rubbish itraconazole formulation. So we're absolutely, it's absolutely critical that we have some faith in the compounds that we are using are the same in Vietnam as they are in the United Kingdom. And the problem is, it's not the generic, but it's the data and the accessibility to the data that gives us the faith of everything else that follows in terms of the clinical trial. Right, so just to finish now, amphotericin B. I see some old Gilead warriors in the audience. I guess Emma and Johan too. You can't see this, and maybe that's a good thing, because when you look for amphotericin preparations in India, of course there is an ambisome there. But it's not ambisome as we know it in the UK. And of course, all of these preparations, as I suggested, are not covered by would not be. They don't count as the biowaiver legislation wouldn't be able to mean that these compounds were shepherded into the United Kingdom or the EU. They have to have their own actual clinical studies to get licensed, because you can't use PK for the bridge. Well, that's more of them. These are all Indian preparations, of course. And of course, everyone in this room knows that the PD, the pharmacodynamics of these various formulations of amphotericin B are completely distinct, and therefore can't be easily or readily bridged. The clinical data from each of these compounds can't be bridged into the clinic. So, in conclusion, Tony, we have very stringent regulation in the EU, and that provides some comfort for the use of generics, I think. For global problems, generic medications is an interesting problem, and there's this fundamental assumption that is often not critically appraised, especially by me, that the drug is the drug, and perhaps actually there is an opportunity here for the community to reflect upon this a little more than I have seen done in the past. And with that, I'll stop, and thank you very much for your attention. Can I sit down?